My name is, if you don't know me, I'm Peter Kolb. I'm the MSc Extension Forestry Specialist. Lived now in Montana for 18 years. I've been working in the Northern Rockies for the last 30 years. My background is actually tree physiology with a smattering of genetics and silviculture, so kind of a broad-based arena, but really Northern Rockies has been my area of interest most of my professional career. And so there's different topics that I explore at different times, and genetics is one of those things that has, our knowledge of genetics has grown phenomenally in the last 10 years with modern electronics and the ability to map out genomes and things like that. And with that knowledge, some information that I think is worth talking about, because historically genetics has been used to breed for or select for characteristics and traits that we really wanted in something. Taller trees, bigger cows, fatter pigs, things like that. And it's worked pretty well with us, for us, but there are trade-offs when we do those types of things. And so why trees do what they do and the difference amongst species and what we're really selecting for, I think, is worth discussing. And a lot of this is going to be fairly theoretical in basis, because doing genetics work is very expensive and extremely time-consuming over periods of time. To date, the entire genome, which means the entire genetic code, has been mapped for 2 tree species. Loblolly pine in the southeastern United States and Norway spruce in Europe. Norway spruce is the bread and butter tree over there. Originally, it may have occupied about 10% of the cover type in Europe. It currently occupies about 40% of the cover type of forests in Europe and is responsible for about 80% of the wood volume produced over there. Produces white wood, straight grain. It's usable in a lot of different applications. And loblolly pine, of course, was very important to the paper and pulp industry in southeastern United States. So there's been a lot of work on that species as well. So our species here in the Northern Rockies, we know some things. White pine blister rust work, and we used to have a tree nutrition or a tree genetics co-op that looked to grow taller growing, faster growing trees and things like that. But really not a whole lot of the kind of work that might be pursued in the future in this arena. So I'm going to give a pretty quick overview of some core genetics and then try to relate that to how we manage our trees. And whether we know it or not, or like it or not, we're doing genetic selection every time we do any kind of manipulation of a forest. So many of us as forest landowners, and I own 20 acres up in Evero, I've harvested a couple loads off of it. I have actually the map down in the snack room of the property is my property. I have 12 management units on 20 acres, which some people might find laughable, but I have a wide variety of species on it. I've got moose and bears and bobcats and a lot of neotropical birds, and they really like the diversity that I have on the property, and so I manage for diversity. And I wrestle with bark beetles like everyone else does. And the genetics issue is kind of an interesting one when you think about pests and pathogens and growth rates. So when we're out here selecting trees, and whether we leave that one or leave that one, and or whether we do it by spacing or not, we are having an impact on more than just what's growing there. We're having an impact on the genetic resource that's out there. And most of us, I believe, when we are managing our forest, what we leave behind, we want to grow and live. We don't necessarily want it to die, unless we're specifically managing for snags on a property. So, when you look at any landscape, and I do this every time I'm out in the forest, and I look at this and I go, okay, so why was this tree hit and not that one? Why were these 2 hit and not the ones next to it? And sometimes the answer is, well, they haven't gotten there yet, and I come back the next year and those have been hit as well. But there's a lot of diversity out there, and of course, species is the easiest one. Doug fir beetle isn't going to hit ponderosa pine, but there's age class diversity, and there's a lot of genetic diversity that's out there, hopefully. And so when we take a stand like that and we thin it for fire hazard reduction or tree vigor and health, what tree we decide to leave and which tree we decide to take is the essence of forest management. And I think most of us, myself still at this stage, sometimes debate and puzzle over which tree we're going to leave. And on my 20 acres, right now I'm preparing to harvest another load of logs off of it, and I've been agonizing over which trees I'm going to cut now for the last 6 months. I have that luxury on 20 acres because, I like the money I get from it, but there's a lot more at stake there. I mean, and so once you take a tree down, you can't glue it back up again. So what spacing, do we go by spacing if I'm following a 20-foot prescription? Am I going to leave a tree because it's in the right spot, or am I going to leave it because it has certain characteristics? And how do I trade that off? So for example, here's a ponderosa pine with a very nice, thick, full crown. That crown means, and all that leaf area means, it has growth capacity, photosynthetic capacity to produce sugars that are going to produce wood and allow it to grow bigger and taller, and hopefully defend itself. Whereas something that has a pretty poor crown, very thin, sparse needles, has less capacity to produce energy and therefore, theoretically, less ability to defend itself. And so the question then ought to come up is, okay, does this tree look good because it has all the space it needs? This one looks poor because it was crowded and it's lacking resources, and will it recover from this? On the other hand, sometimes, has happened to me, I leave what I think is the best tree, and the next year mountain pine beetle comes and hits it. And then I'm madder than heck because I took the trees out from around it. And I'm wondering, okay, how can I up my odds that that doesn't happen? Because if I leave it, I want it to grow. I don't want it to feed beetles. And this applies to every age class, whether it's younger or it's older. We're always looking at it. And when you look at any group of trees, you're going to see differences in characteristics on those trees. Some are going to be a little taller, some are wider, some are narrower. And why is that happening? So what we see is the product of the environment interacting with the genetics. Okay? And every population has some level of genetic diversity built into it. What we lack in ability right now is to sample this tree, and see if they're genetically different. Well, we know they're genetically different because of just natural variability in the population. But is the crown shape and the potential for it being hit by a pest or a pathogen, can we determine that? Okay. Now in humans, we have mapped the human genome. And you can enroll in a program called 23andMe and some other ones, and send in a saliva sample, and they'll map your genetic background. And they used to also tell you what your predispositions are for whether it's liver cancer or strokes or heart disease and all that kind of stuff. And the USDA has prevented them from doing that because they're afraid that us dumb people, once we read that we're predisposed to something, we're going to change our pattern of how we do things, which is the whole point behind it, I thought. Anyways, but nonetheless, I guess we can't be entrusted with too much information. It would be nice at some point in time, I hope what the future may bring is that we have a little sampling kit where we can sample this tree and we can determine whether it has markers for certain predispositions. And we can start using that as a selection process. And I'll get into that a little bit later on. Yes?
I'm just curious, why, what you just described is pretty basic stuff in genetics now.
Right.
Why hasn't this been done? Is it just money? I mean, certainly there's an interest, right?
Well, yes and no.
It's not that expensive, really, to do.
Well, it is very expensive to map a genome. Okay, consider that the human DNA has about 3.3 billion base pairs. Okay, and what makes you and I different is about perhaps 220,000 different proteins or genetic codes on there, and it's getting cheaper, certainly. I mean, when I was, and you seem to have some knowledge on this topic, but I recall 20 years ago, some of my friends who were graduate students were working on some of the real basic stuff on this stuff. And one, the variability was killing them, and two, the testing was very difficult. But Yeah, it is. It is
I mean,
getting.
then it was very expensive.
Right.
Now it's
Right.
just not.
Well, right. I mean, for $100, I can send in a saliva sample and find out, you
It's
know.
more than that for doing that, what you want to do.
Right.
But, yeah.
Well, I hope it will get to that point. Like I said, right now, the Norway spruce genome was just finished mapping last year. Okay, and loblolly has been mapped. No other tree species have been mapped.
This is just because of the economic value, that's it?
Well, yeah, I mean, if you're going to do it, someone has to pay you for it, I suppose. You know, and yeah. But as we get better at this, and there's, you know, on one hand it is fairly basic and simple, but on the other hand, there's a lot of details that aren't figured out yet on that. You know, specifically, Norway spruce has a genome that's as big as ours is. But half of that genome isn't used. I mean, it's what they call, they have a certain name for it, trash genes or whatever, or leftover genes from, you know, conifers have been around for 80 million years. Okay, and over that 80 million years, there have been, the different species have splintered off of it. But for some reason, unlike humans, spruce keeps these duplicate sequences of genes. And nobody understands why. And I'll get into that a little bit more. So there's what may seem simple at face value actually gets complicated in a hurry, and as they say, the devil's in the details. And how much you interpret from what you know gets kind of scary as well. So, you know, and it's not only mature trees when we're doing a selection process, then there's also the regeneration. And are there characteristics in regeneration that we should be looking at and looking for? Can we start looking at this when they're little? Do they need to be bigger? And certainly insect or specifically disease resistance is one of those things that we may see more as the tree is older. Gall rust in ponderosa pine, which isn't that common, but certainly when you get into a population, you'll see some trees are just covered with gall rust and other trees are not. Okay, so there's a selection process going on there and some indicators as well. And so I'm going to give you a kind of a basic overview, and you know, if you're knowledgeable in this topic, I apologize that it's going to be pretty basic. But there's a lot of terminology in genetics that can be confusing and conflicting, and so I want to go through some of that. I think it could be simplified a bit more. So heredity is what we're really talking about in genetics, the passing of some traits from one generation to the next. Okay, and so you know, what's a trait? It's a specific characteristic that's unique, that affects the way something looks, the way it functions, and that can be inherited. It's not necessarily just, we go back to the Cold War after World War II, and the Prime Minister of the Soviet Union, Khrushchev, made a fatal mistake in their agriculture because there were two different theories of genetics at the time, one which was disproven but he happened to adhere to. And now one that he adhered to is that when you do something to an organism, it gets ingrained in its genetics. So in other words, if you cut off the tails of mice, those mice will produce offspring without tails. And he based their entire agricultural system on it, which failed miserably as a result of it. Versus, okay, what's truly inherited, what's genetically based, and what's environmentally based. And when we're talking about trees, that can be really, really tough, well, in humans as well. So, you know, what you see, term phenotype, is a combination of the genetics and the environment acting on that genetics and the interaction of the genetics with the environment. And I'll be so bold as to add a few things onto that even. So here behind us, this is actually on Foothills Road, right behind Cabela's, where someone planted a bunch of Rocky Mountain blue spruce for privacy. And here's a common question I get: I planted blue spruce and now they're turning green. What can I do about it? Okay, so here we got, these are all the same planting, and here we have a green one. Okay, well, what the blueness is basically the wax that's on the needles curls and refracts blue wavelengths. Okay, blue wavelengths a little shorter than other wavelengths, and gives it that blue sheen. Well, on some blue spruce, as they age, that characteristic disappears, and the wax is more of a smooth coat on it, which then gives it the green appearance. Okay. Now, can that be modified by environmental stimuli? Probably. It's not proven, though. Certainly, people have observed that when they plant blue spruce in certain locations, suddenly they lose their blueness, and other places they don't. So, you know, here is a genetic thing. And horticulturally, blue spruce have been propagated from certain individuals that had that curly cuticle material on them, and so that's now a highly desired ornamental because of the blue color. Then there are other things. In trees, historically, selection has not only been based on color for ornamental purposes, it has been on growth rates and cold resistance, because we have tree zones. You know, if you do any kind of horticultural work, you know that we're in zone 3 or zone 4 here in Missoula. If you go out into Great Falls, you're in zone 5, zone 5, or zone 4, zone 5, and that has to deal with when it gets cold and how cold it gets. Okay, zone 3 is cold tolerant down to about minus 10 degrees. Zone 4 is cold tolerant down to minus 20 degrees. And so this winter, cold tolerance in trees is basically triggered by day length. When days get a certain shortness, trees are programmed to start preparing for cold weather, which means they produce lipids and sugars in their cells to keep the water from turning to ice, because when water turns to ice, it expands and blows little cells apart. Okay, and there is genetic variability in that. So zone 4 trees are more cold tolerant because they have that genetic trigger that tells them when to go dormant. Well, this November, early November, as you all experienced, we had an almost record cold, minus 10 here in Missoula, and it caught that one. Okay, it hadn't hardened off completely. Now, can you modify that with environment? Absolutely, because if we get to it, I have a slide in the back on my own property. Some grand fir were hit by that, the new needles, and some were not. And particularly those that were growing underneath the cover of another tree were hit particularly hard, because that thermal layer, they hadn't hardened off completely. I have other grand fir where only half, a part of the tree got hit and the other part didn't. And it's not necessarily microsite, it's because those specific branches had not hardened off when other ones had. There's a recent study that came out on eucalyptus trees growing in Australia, where the eucalyptus tree has this unique ability that every branch has different programming, and so will produce different levels of terpenes and phenolics in it. And they're suspecting its adaptation to severe defoliation by koala bears and other organisms, that the bears will like some leaves but not others, and that way the tree will never be completely defoliated. So pretty cool that, you know, so there's all sorts of things that we're learning nowadays with modern technology. And the bottom line is, as we all suspect, it ain't as simple as we would think it is. Now, again, as I mentioned, historically, when you buy tree seedlings from a nursery, most of those seeds come from select sources, seed orchards. Okay, and I was involved with this work 30 years ago when Champion was making its seed orchards around. And what they would do is they'd go around the countryside looking for what they called superior trees. So you look on a landscape and, you know, what would you face value call a superior tree? Well, it's one that got big and tall faster than others. Okay? And so the big tall trees from all across the countryside, they'd go up and they'd snip off a branch, and that branch was grafted onto a rootstock of something else. And those are now tall trees producing cones from that particular tree, even though the rootstock is different. Plains, Montana is the one I was involved with directly. And the concept there is that this tree is bigger than the other ponderosa pines around it because it has the genetics for height growth and volume growth. And that when we get that genetics from all across the region and we put them on a seed orchards, they will cross-pollinate each other, and there will be a high probability that their seed and seedlings will have those superior growth characteristics. Now, mind you that Champion and Plum Creek and all the companies that were looking at this were looking at growing volume because they're in the business of growing wood and producing wood products, okay? And one of the things we certainly have learned about genetics is that when you select for some things, you might be selecting against other things, okay? And so, and it's gotten even more complicated than that now. So, again, well, originally with all breedings, how do we keep track of things? We create these pedigree charts where we have a male and a female that have certain characteristics, and we track how those characteristics might be passed on to offspring. It might be dark hair, brown hair versus blonde hair, blue eyes versus brown eyes, things like that. And then we can track how things are passed through. And it tells us a little bit about the genetics. For example, it might be sex-linked. So males have certain characteristics and females have other characteristics. So here the male might have, you know, what's represented by a vertical line here, whether it's blonde hair or blue eyes or who knows what. And that trait is passed on only to the males, and the female characteristics are only passed on to the females, and that's associated with the XY chromosome. So that's really simple stuff and can be mapped pretty quickly. If you start mapping multiple traits, might be eye color and hair color, you might find that hair color, dark hair, can be passed on to all sexes, offspring, all genders, and whereas, you know, the other characteristic stays associated. And again, this is pretty simple, and you know, then we can get even more complicated where we don't necessarily see a pattern. Some of the kids, offspring, whether they're male or female, get these characteristics and others do not. Okay? And so when you consider that a human genome, and I'll explain what base pairs means, you know, that there's a variability of 3.3 billion combinations, okay, on our DNA strand. That's a number beyond comprehension, you know, other than that, that's huge, okay? And that there's a certain similarity on all higher organisms in their genetic code, in that we may share something like 60% of the same coding across all higher level organisms, which, you know, seems like a lot, but when you think that what we have in common, all of us in the room, we have probably about 99.9% of our genes are in common. That still leaves 220,000 different proteins that differentiate us. So it's still a lot. You know, these scales of magnitude are something that are sometimes hard to comprehend. And so we might come up with these types of pedigrees that's used in dogs and chickens and horses and horticultural purposes. The Westminster Dog Show just finished, you know, and what is a beagle won it and all that. So you have these pedigrees with these certain characteristics that we hopefully pass on, you know, nowhere more prevalent than in horse racing, you know, where this year's Triple Crown winner, all of his you know, is high valued because hopefully those characteristics will be passed on. But a lot of times they aren't, because there's a lot of variability into that. And we can't talk about genetics without giving credit to Gregor Mendel, who is an Augustinian monk who started all this stuff by noticing the differences in pea flower colors, pea shapes, pea pod colors and shapes. And he started all of this stuff, and he kind of came up with the concept of dominant and recessive genes, or traits within genes. And simply to explain that is we all have what genetic characteristics are typically passed on from two sides, the male and the female side, okay? And in certain characteristics, there are what are called dominant and recessive traits. So the dominant traits are that, you know, if you get it, that's what it's going to show, and the recessive means you really have to be purebred in that trait in order to show it. And even though I don't know what's true, I use this example of common spring flower. Here's glacier lily. You look at glacier lilies, 90% of glacier lilies produce one flower. Okay? But occasionally you'll find some that produce 2 or 3 or 4. The most I've found is 5 on one seed head. Okay, you go, well, why is that? Okay, is that one grown on a nuclear site or what? You know, and this is the genetic variability that may exist in everything. The advantage of being coded to produce one seed head means that this plant and these leaves would put all their energy into this one flower, which will then produce X number of seed, and the chances are that that seed will have enough energy and be viable to produce offspring. Okay? The disadvantage of having one flower is along comes a deer and bites off that flower, and this individual plant is just out of luck. Okay? So what if I produce multiple seed heads? Okay, if one flower goes bad, I still have these as backup, which may be an advantage. Or the disadvantage is if I'm producing 5 times as many seeds, I may not have the energy to produce viable seeds in all of them. Okay, so it's going to require more energy for me to make this work, and chances are that I'll have 5 flowers, but none of them will produce a viable seed because I don't have enough energy to fuel 5 flowers. Now I'm speculating on all of this because this is very rare and this is very common. And so those characteristics that give an individual or a species an advantage are the ones that are propagated, are the ones that produce offspring and therefore are most common. And this might produce an advantage under certain individual circumstances. Okay? Now, the other question might be, well, how do I know it's genetic and it's not environmental? Okay, I joked that that might be on a radioactive site. Well, maybe this one is growing on a site with a lot of nitrogen in the soil and it has more energy to do that. And the answer is, I don't know. Okay, separating out environmental impacts from genetic impacts are very, very difficult things to do and can only be done by controlled breeding environments, greenhouses, etc. Yes?
Would that be an expression of epigenetics?
Interaction with genetics with environment?
Yeah.
Yeah, it certainly could be, and I'll talk about that a little bit later on. Okay, but in the simplest form, this is kind of just trying to explain how selection and genetics works across our plants and trees and people as well. So a dominant trait is a trait that's always expressed or shown. Brown hair, brown eyes, right-handed in people. Okay, so if one of the parents is homozygous for that, well, I get into that, or it has that trait, more than likely most of the kids will have that trait. A recessive trait is one that's covered up or seems to disappear. Blonde hair, blue eyes, left-handedness in humans. So traits come from these things that come from both parents, called the genes. Okay. So a gene is a location on a chromosome. This is a map or a picture illustration of the human chromosome. Okay, there's 23, and this 23rd one is, whether this was an X or Y determines gender, whether you're a boy or a girl, and there's certain things linked to that. So a gene is simply a chemical sequence on a specific location on a chromosome. So, on this chromosome, there's a strand of DNA that has a certain sequence on there that is a template for a certain kind of protein, and that protein is used as a building block for a certain characteristic on us. This is what it boils down to. Humans have 23 chromosomes. Most tree species have 24, though some, such as gray leaf willow, have 152. Okay? Trees and plants can get weird in a heartbeat. Okay? And they've been around for a long time. So basically, if we deconstruct a chromosome, what it is a tightly packed, specifically packaged DNA strand. Okay, this is that twisted ladder, the double helix. This is a DNA strand, and in that DNA strand, there's 4 base pairs. Okay, these are core parts of our DNA. It's adenine, guanine, cytosine, and thymine. And let me back up here. Oop, other way. And these make different combinations, creating the DNA strand. Okay. As I mentioned, the human genome is estimated to have 3.37, I think, billion pairs on it. Okay. And each sequence on here is basically a template for the precursor to a specific protein. And that precursor or enzyme or component, and that protein is then used to construct a certain part of an organism. Your fingernails, your hair, the membrane on your eyes, etc. Okay, so that's kind of how it works. It's not too different in a simplistic form of having a template for different pieces of wood that you can use to assemble to make a chair. Okay, each piece of that template makes a part of it, and then the assembly is what happens in the physiology of your body. Okay, so different maps of how it looks and how these proteins might be forming, or precursors to proteins might be forming on this chemical template that then get assembled into larger molecules. So basically DNA is the instruction code that the genes use to form traits. So there's a master code, if you will, and the genes are the short pieces in that DNA. So to make different parts in a body. Okay, so again, if we look at a illustrated version of this, gene 1 might be a certain sequence on there, gene 2 might be a different sequence. They can be different lengths, different complexities. Some genes are enormously long, making super complex proteins and molecules. Norway spruce has 29,000 functional genes. Yes?
Do genes have codus, like rest spots that say this is the end of the gene?
Yes. And this is how a lot of the modern gene transfer work and gene splicing occurs, usually using a virus. Because the way viruses work is they insert their own form of DNA. It's not true, not DNA as we know it, but it'll splice itself into another organism's DNA, and then when that cell reproduces, it reproduces the virus as well. And so these viruses actually have been used. You can insert them with a specific section of DNA, and you can use that virus to splice something else's DNA into another organism. Okay, those are called genetically modified organisms, which the European Union has banned. Okay, and I'll get to that a little bit more. It gets a lot more detailed and complicated that is beyond my knowledge, and that's why it's a science unto itself. Okay, but the point is that you think about when I say 29,000 functional genes. Spruce is kind of interesting. Like I said, trees are weird because about half of spruce's DNA is not utilized. Okay, it's considered an archaic remnant. Nobody's sure why. Okay, but half that DNA is just there, and there's genes that just aren't expressed or aren't utilized, and nobody's sure, or they're replicated. There's the same sequence over and over and over again, and nobody knows why. Okay, now again, conifers have been around 80 million years. And so the different species, pines, spruces, etc., are all splintered off of that common core of conifer. And so the thought is with spruce being one of the oldest, it's carrying all this old baggage with it, and because it just doesn't have the ability to shed it. So, but again, this is stuff that people ponder about.
So would that maybe come into play as like a memory for the tree going back to, let's say, ice ages? So if they need to pull that information, they'll still have that information.
Well, that's an excellent question, and if you have patience with me, I'll try and answer that. Okay. Now, the important thing on all this stuff is to remember, memory is is a good term, but we don't use our DNA cognitively. It would be nice, if we could, but this is programming that happens.
Right. It's not a
muscle
memory, but
muscle memory.
Yeah,
It's not cerebral.
Yeah, and it's important to remember that because sometimes we think of we anthropomorphize stuff and anything is making a conscious decision. But there's actually a lot to your question, and it's an excellent question. But let me cover some of this other stuff first, and then we can get to the fun stuff like your question. Okay, so there's 2 genes theoretically for every trait, one from each parent. And that gene pair is called an allele. There's a lot of terminology. I think geneticists could simplify this down a little bit. But anyways, in trees, one gene comes from the pollen, the other comes from the ovule in the flower. Okay? And they're expressed in different ways. And the terms we use is homogeneous or heterogeneous or heterozygous. And so what does that mean? It means that if an individual trait gets a part from each of the parents. Okay, there's 2 parts to it. If those parts are the same, we call it dominant, then they're described with a capital letter. So say the gene for tall is capital T. If the gene it got from the mother and the father are both this dominant T, then it's considered homozygous for that, and they'll all be tall. Okay. Likewise, if the mother and the father both have a short gene, and that would be then homozygous for short. Now the other part of this is what's called a heterozygous or mixed, and that's when if the mother has a dominant gene and the father has a recessive gene, then it's described as heterozygous, and the dominant expression is what you see. Okay, so in this case where it has both, that individual will be tall. Okay. And the only way, if it's a recessive, the only way it can be short is if both of those codes those inherited traits are for short. Now, the way we historically do this is called a Punnett square. So we have the genes of both parents, okay? So if the father is heterozygous, has a dominant and a recessive, and the mother's heterozygous, has a dominant and a recessive, then basically 3/4 of the offspring will show the dominant trait, and only 1/4 statistically will show the recessive trait, just the combination of how these things combine, okay? And that way we can use that, like Mendel developed this technique to predict what percentages of offspring will have what specific traits. So in this case, the P, where we're looking at 2 different characteristics. One is greenness of the pod, shown by a by the G. So the big G is green, the little g is yellow. And then the color of the seed, the P itself, where the dominant is yellow and the recessive gene is green. Okay, so separated by a big Y and a little y. And so we do the math on this. You can see that the majority, 3/4 of the offspring of the pods, will be green, and 3/4 of the seeds will be yellow. However, in different combinations. Okay, they aren't necessarily connected to each other, depending on what's dominant or recessive on there. And so, this now gets back to how do we look the way we do, or why do we look the way we do? Is that offspring are similar parents, but they will be different because they have those combinations of the parents, right? So, and this is known as the phenotype. That's the term for this is what you look like. So if we go back to this, there really are 4 different phenotypes: green pea, green seed, green pea, yellow seed, yellow pea, green seed, yellow pea, yellow seed. Okay, this would be the double recessive, and you can see that the more complicated it is to get the pure recessive becomes less and less of a probability. So only 1 out of 16 possible combinations will give you both recessive traits for both these characteristics. Okay, so and how does this apply? Well, this is real life. My nephew was born deaf. He was born deaf because he has my brother and his wife both have a very rare recessive gene stemming from Eastern Europe that prohibits the full development of the cilia in the inner ear. Okay, and so my nephew got unlucky. Now he's got an implant and is about as normal functioning as any of us are now, modern technology. But chances are, he was told that chances are if you have another child, it probably won't have that because of the rareness of both of those things coming together, because it's not as simple as a combination of different genes in there. But this is how we use these things to predict future offspring and how things happen. So the genotype is the genetic makeup, and there's lots of different ways that this can happen. So for example, if one parent is homozygous for the dominant gene, all the offspring will show that dominant trait, even if the mother is heterozygous for that characteristic. Okay, so what we see is all that dominant trait, right-handedness. On the other hand, if you have both parents are heterozygous, then 1 proportion and 1/4 will show that left-handed recessive trait on there. And we can go on to other types of combinations. So here both are recessive, so naturally all the offspring will have that recessive trait. Here, if one parent is dominant and the other is purely recessive, again, only the dominant trait will be shown. Now, this assumes, for simplicity, that this one trait is only controlled by one gene, or the gene pairs, okay? And so we know, of course, that a lot of traits are controlled by multiple genes and combinations thereof, and that's why very few of us have pure brown eyes or pure blue eyes, and we have everything else in between. So incomplete dominance is then also when neither gene determines a trait in a dominant way, and you end up with something mixed. So a red flower or rose mixed with a white rose will produce a pink rose, okay? At least that's the theory. Now that doesn't, of course, always happen, and so I know there's a lot of breeders that try to breed different things by figuring if I mix these two, I'll get a combination of the two. And that doesn't happen when one of the alleles is dominant and the other is recessive. Now, how do things change? Okay, every population has all this variability, and as I mentioned, the human genomes, I mean the difference between him and I, or him and me, is that 220,000 different potential proteins that get produced by our different DNA. Okay, how do those different ones develop, and how does a population do it? Well, number one is mutation. Okay, that's when something in that strand of DNA changes, either when it recombines, when an offspring is produced, one of the sequences happens to break off or fall off, or isn't replicated, sticks together, something like that. Now the majority, something like 99.999% of mutations aren't viable. They don't really continue on. Okay, either they're detrimental to the organism, it'll die, or it's just not something that necessarily shows any kind of advantage to move forward. So then we have natural selection. This is an evolutionary process where, say a mutation occurs, where ponderosa pine develops an ultra-thick wax layer on its needles. Or rather than producing 3 needles per fascicle, it produces 2 needles. The difference is that the 2-needle one has less surface area than the 3-needle one, and therefore loses less water in a dry environment. Okay? The one that only has 2 needles in a dry environment is more likely to survive and produce offspring. And those offspring are more like the ones, not all the offspring will necessarily have that 2-needle, 3-needle thing, but those that have the 2 needles in that hot, dry environment will tend to survive more often and reproduce, and therefore that specific genetic characteristic becomes a greater percentage in the population. Okay? So, which may be why in eastern Montana we have 2-needle ponderosa pine, where western Montana it's 3-needle ponderosa pine. It's speculation, okay? But, you know, and sometimes it doesn't have to necessarily have a pressure on it. It may just be that has neither a good or a bad effect, but it's there and it just persists in that population. Okay?
An example of that would be in the Congo, people developed sickle cell anemia, which is considered a disease, but they reproduce and live longer because malaria doesn't affect sickle cell anemia people. So that population grows and flourishes because they're more resistant to malaria.
And that also brings up, it's an excellent point, one that's often given for genetics, brings up another important point, that in this selection process, that selection process only applies to reproduction. So it doesn't apply to longevity necessarily. Longevity may play a role if you remain reproductively active. But as long as an organism with this new thing can reproduce and its offspring can reproduce, then this becomes an advantage. Okay? And so, you know, that's why we look at sometimes genetic advantages like sickle cell anemia is for reproductive rate. It is not for longevity. And so the downside of having sickle cell anemia is you survive malaria. The downside, or that's the upside. The downside is when you're 50, you'll probably die, and you won't make it to 80. You know, but there's, you know, no necessary advantage there, unless having grandparents around gives your offspring a greater probability for surviving. So you can play all of these kinds of games on how this works. And you know, natural selection, boy, it gets misused a lot in what's considered better as well. All right, well then we have what's called migration, and that is when, say, you have a mutation in one population, and its offspring or its genetic coding, in the case of trees, pollen, floats into another area. Okay, now you're getting something new through migration. Okay, so that's another way that gene frequencies can change in a population. And then finally, there's called random genetic drift, and this is probably very, very important in tree species in the Northern Rockies. Random genetic drift is when happens usually in small populations. And so just by happenstance, say a whole population is wiped out and only 2 people are left alive, or 2 trees are left alive. Okay? And they breed, produce offspring, and as a result just of happenstance in that breeding process, certain genes are excluded. and then that population grows with fewer of this typical type of gene, and that's called drift. It typically will only happen in small isolated populations. If you have a large population that's interbreeding, you won't see that, and the gene frequency change stays pretty similar in that population. Now, this is probably important in the northern Rockies because our mountain ridges go north-south and our wind patterns are west-east. Okay? So we have a lot of geologic barriers to pollen dispersal, okay, in the form of mountain ranges and mountain ridges. To the point, say, if you look in the Bald Marshall where you have those really deep rocky ridges in there, every valley has a small isolated population that might have been subject to genetic drift, and therefore that population might start becoming genetically unique. Okay? You can find that in other organisms, you know, where say with cutthroat trout in one population they got 3 spots on their head, and the other population they got 10 spots on their head. Are these genetically unique populations? Do they do we need to protect them separately because they're genetically unique? So where you differentiate between a species, a subspecies, a trait, an ecotype becomes pretty dicey in there, particularly as we want to protect uniqueness and diversity and things like that. And that remains a pretty good debate on a lot of things. You know, what is a viable grizzly bears? What's a viable genetic population where you have enough genetic mixing occurring to keep that population robust and healthy? And we'll get to that here shortly because that also applies to trees. Now, selection can affect gene frequencies pretty rapidly. So, you know, this is off of an actual biological population where there was a certain characteristic in their genes that in a broader population was just fine, but in a small population hampered its ability to reproduce or have viable offspring. And so in a matter of generations too, with every generation the frequency of that gene becomes half of what it was before, and so that gene frequency can become almost non-existent in a population if there is some selective pressure against it in a specific locale. Okay, so that, you know, things can change. And then there's this thing called heterozygote superiority. Okay, now sort of one of the assumptions in most of these combinations is that when you have a dominant and recessive gene, the dominant one is the only one that's functioning, which isn't necessarily true. It is the one that is dominating the traits that you see. Okay? The recessive gene may still be functioning because remember, a gene is simply just a template for a protein. Okay? So it still may be producing proteins, but at a smaller amount than the dominant one. And so what has been found is that when there is this diversity in the genes, not just the dominants, but you have the dominant and the recessive across a broad range, a broad spectrum of that genetic code, that those individuals tend to survive better. Okay? So a classic example would be dogs. Okay? Dogs have a common ancestor that was something like a wolf or a coyote. Okay? Now wolves and coyotes have kind of been selected on their own lines, right? But it's something like a wolf or a coyote, all right, looking type of animal. And through inbreeding them, we have looked for specific characteristics. You know, so these mutations that are rare that create gigantic animals like Irish wolfhounds, or tiny animals like Chihuahuas. Thank you. Okay, so we have through inbreeding managed to select out all these different characteristics, traits. But in a general sense, as a generalist, if you throw any of these animals out in the woods to survive, they're going to die because they don't have all of the characteristics necessary to adapt to a broad range of circumstances. And I like using coyotes because it's probably one of the most adaptable canids in the world. They've adapted to survive in downtown LA and live up to the Arctic Circle, okay? And they have this tremendously broad genetics within them, this tremendous heterozygosity, because when they move from one area to another, they might produce 10 pups, 8 will die, but 2 will survive because they have the combination that fits that certain environment, okay? That's called genetic diversity. Now there's also this other thing called phenotypic diversity, where by having that broad spectrum of genetics, they have the ability within them to turn, or stimuli may turn on and off certain genetics in them. So their 10 pups won't die, but pigs, prime example. Domestic pig doesn't have hair, does it? A little bit. But you take domestic pigs and you throw them outside, and within 3 generations, they're all hairy as can be. Okay? Because the environmental stimulate certain genes to become active and grow lots of hair. Now I've tried that. I haven't worn a hat for years and it hasn't worked on me, so maybe I'm not related to pigs. That's a good thing. Okay? But so there's different mechanisms that will express genes. And so this heterozygote superiority is something very interesting to think about, and there's some recent data and research out that in trees really points to this. Okay, and that's the same type of thing of new mutations. Now, so what does this mean about trees? And over the years we've had all of these genetic programmings. This is the Forest Genetics Resources Working Group out of the Northwest that's looking to collect, exchange, and disseminate information about forest genetic resources. And this is one of many groups worldwide, but this stuff really got on my radar screen. I spent 6 months working with some colleagues in Germany. And as I mentioned earlier, Norway spruce is this very important tree species in Europe. Historically, it was 10% of the cover. Now it's 40% that accounts for 80% of the wood production. However, what geneticists there noticed is when we, let me just skip through this real quick to get to a slide that I want to get to. Sorry, I put way more on here than I need, but I'll get back to some of this. Come on. Okay. So what geneticists there noticed is on their Norway spruce is very susceptible to Ips typographus, which is a bark beetle, similar to our mountain pine beetle. Only has 3 generations per year. Mountain pine beetle only has 1. So it can eradicate Norway spruce off of a landscape in a heartbeat. Okay? But they noticed that some trees were not getting hit. Okay? And you'll notice that if you've looked at mountain pine beetle outbreaks all across Montana, 5 million acres. Typically, there's 15 to 20% of the lodgepole pine that stays alive. Okay? And you ask yourself, why is that? Okay? And the theory in the past is, well, there's so many pheromones and beetles are pretty stupid and they get confused. Well, I don't buy that, because when I've been out on acreages where the landowners spray their trees with 7, every tree will have a pile that deep of dead mountain pine beetles under it. Millions and millions, okay? So during an outbreak that the beetles are just missing trees, I don't buy. There's so many of them out there that, they're going and sampling every tree constantly. So what I suspected is that, and still suspect, is that some of these trees have a combination of terpenes they're producing that make them not very tasty to the beetles, or even toxic to them. Well, the interesting thing is, in Norway spruce in Europe, they found the same thing, that a certain percent of trees are just not attacked by Ips typographus. So they started mapping the genes and looking for heterozygosity by testing for specific allozymes that are in the tree. And what they found is that the trees that are not hit have a much greater degree of heterozygosity than the trees that are hit, and the trees that are hit are typically fairly homozygous. And the other thing is, they found an interesting correlation on spruce, is that the spruces that were growing tall, fast, and putting on the most volume were the most homozygous and the most susceptible to being hit by a bark beetle. Now this makes sense, because basically, historically in forestry, what we've been selecting for are trees that grow tall and big quick because we wanted wood, right? We have been breeding the Irish wolfhounds, okay? So we've been selecting for certain characteristics and potentially selecting against other characteristics that are out there. And so they tested Norway spruce all across Europe. In Germany, most of them were planted, and they were planted from select individuals, just like we did. Heck, our genetics breeding program is based on European genetics programs. And so over the centuries, they've always been picking the biggest, tallest Norway spruce, and those are the ones they collect the seed from, and those are the ones they plant. And so they finally found some Norway spruce in the Carpathians that have this broad genetic variability in them, a high degree of heterozygosity. And so they have a program now where they're trying to breed heterozygosity back into the trees. And so there's this interesting correlation that if you're selecting for the biggest and the tallest, you're selecting for the tree that has the greatest homozygosity and therefore the least ability to adapt to a changing climate or to defend itself against pests and pathogens. So, what does that mean for us? Well, okay, if I owned a lot of lodgepole pine, well, gosh, I want to tell another part of the story here before I go to the next step. I'm sorry. I'm going to rearrange this whole thing. Okay, so let's go to this part here. This is another study. Has not that much to do with genetics, but it has to do with species origins across the Northern Rockies. And so this is a lake sediment study, and it was done for most lakes across western Montana, where they did cores of the sediment out of the lake bottoms. And every spring, mud and stuff flows in there, and with that is pollen. Okay, so by aging the cores, the layers in there, you can go back 13,000 years. This is the time scale on here in the history of western Montana. And within that mud, you can count pollen and find a statistical number of pollen of different species. So what I want to draw your attention to on this one is pseudotsuga and larks. Okay, this is Douglas fir and larch. Okay, the width of the black band on here is how much pollen was found in the mud. And what it tells you is how predominant that species was across the landscape. So this is from Foy's Lake up in Kalispell. Okay, so out here, what you see is a lot of Doug fir up until about 2,000 years ago. And then there was a lot less, and then you go back, about 7,000 or so, or 10,000 years, and Doug fir and larch didn't exist. Okay? Now, sagebrush is Artemisia. And what you see, until 2,000 years ago, it's there, but all of a sudden, the gray is variability. So 2,000 years ago, you can see that sagebrush was a lot more dominant on that landscape. And what this basically coincides with, I'm going to skip here again. Okay, is if we go back to our climate graph of the last 13,000 years. This is a log scale here, so 2,000 years right there. Okay, the first 8,000 years since the last ice age were a lot warmer and drier across the Northern Rockies. Okay, and this is since the last 2,000 years. This is medieval optimum right in here at about 1,000 years, and then this cool wet, and here we are now. All right, so let me go back now. I'm going the wrong way, I think. Sorry. I had something different in mind when I set this up, and I gave my first talk and go, God, I should have moved those around. So we go back to, here's this 2,000 years ago. Okay, so Douglas fir and larch did not exist as dominant species up in the Kalispell area. And if you look at the rest of Montana, it didn't exist that much in Montana, period. Okay, till 2,000 years ago. Well, 2,000 years is nothing in a tree's lifespan. That's 4 generations of Douglas fir. And if you happen to be in the jarred larch grove up at Sula Lake, that's 1 generation. That big old larch tree might have been the first larch in that drainage as the glaciers, or as the climate got wetter and supported trees. So think about it, that you're looking at maybe the first larch seed that landed in Sula Lake Valley in that big old larch tree. Okay? Kind of mind-blowing when you think about all of this and the genetics that's in there. So now there's this other thing. Remember we talked a little bit about, did we talk about inbreeding? Okay. So if we look at these populations, the Douglas fir and the larch that we see across these landscapes out there originated from tiny little populations. Okay. So this is pinus, Douglas fir, ponderosa pines, and white pine all mixed together. So it's kind of hard to tease apart what was what on there. But let's go now. Oh, I didn't talk about that yet. Well, let's go back to this phenomenon. Okay? Now we have these expansive forests across Montana that originated from small isolated pockets of populations. Okay? So think about that. Let's talk a little bit about inbreeding. Okay? All conifers have the ability to fertilize themselves. Okay? So they produce pollen from the ends of the branches and the cones just a little further back. Okay? And their timing is not necessarily different. So conifers will inbreed a fair amount. Douglas fir is notorious for doing it a lot. Okay. When you inbreed, it means that what you tend to do is lose variability in your genetics. You become more and more and more homozygous, which is why certain things will come out that sometimes are not desirable or are desirable. As I mentioned, most of our dog breeds are a result of inbreeding. You get gigantic dogs by inbreeding, and that giant gene starts to become more dominant, and that's how you get an Irish Wolfhound eventually, or vice versa. The problem, however, is that after 4 generations of self-fertilization, every generation you do this, you cut your heterozygosity in half. So your population becomes more and more and more genetically similar and homozygous. Okay? So now, let me go back.
Take it away from her.
Hey, as long as it doesn't explode, I'm good. You know? We go back to this phenomenon that we're seeing across Montana. 5 million acres of lodgepole pine getting hammered by mountain pine beetle. Okay? And then you think about the origin of our individual species of trees. They came from very small populations, which means, based on all of this stuff, statistics, that most of our trees, and they did this the last 2,000 years. So from 2,000 years, you go from 2 pigs to 1 billion pigs. Is that population of pigs going to be inbred? You're darn right it is, okay? And what you do when you're inbreeding is you're breeding against heterozygosity and diversity. So what may be happening across our landscapes is basically we have these pests and pathogens that, like it or not, are kind of cleansing agents on the landscape, is that they are targeting, based on the spruce studies and all that, all the homozygous and inbred, and leaving those with the greatest genetic diversity behind. And this is often, I've speculated over the last 30 years, you'll see mountain pine beetle or any kind of pests and pathogens often occurring in pockets on the landscape, right? And again, with mountain pine beetle, it's attributed because, the 2 pioneer beetles land on this tree, they emitted this pheromone, beetles are stupid, so this cloud of pheromones, so the beetles hit all that group of trees, which may very well be a mechanism. Well, it is a mechanism. But the other side of that is that when we look on a landscape, pollen does not travel great distances in dry and low humidity. It lasts in a super 80 degrees and 10% relative humidity, pollen dies in about 5 minutes. Okay? It doesn't survive a long time. It survives longer in cool, wet environments. Okay? So when we look at a landscape of trees, what we have, and they found this with spruce in Europe, in the Carpathians, that about every 100 to 200 yards you go, each of these is a related population. Okay, of what's going on there. And so they're even more inbred. And so you have these insects and diseases hitting these weakened populations that have lost their genetic diversity, and what they're leaving behind are those that have, by pure statistics, crossbreeding, have the broader heterozygosity, the broader genetic signature. Okay, this is a selection process. So you ask, well, okay, if this has been going on, then why don't we have mountain pine beetle resistant lodgepole pine? Okay, lodgepole pine stores viable seed for up to 30 years. Okay, what is the primary disturbance that promotes lodgepole pine? Fire. So bark beetles came in here, they did this selection process, has potentially, if the spruce analogy works, has potentially left the genetically superior trees behind. But these trees are not going to have a reproduction advantage because all that fuel in the ground, this is going to burn. And all those seeds that are stored in the serotinous cones of these dead trees are then going to open and repopulate the landscape. And so the selection process that the beetles do is erased by the wildfire that burns through it and kills it all. Okay? Now, with climate change and the theory of warming climate, and some people get incensed by me calling it a theory or even mentioning, or on the opposite side mentioning it, it is a theory. It's a very valid theory. There are some questions that need to be answered in there. It's, like anything, it's a complicated phenomenon. And so I can with no more confidence say it's not that human CO2 is not impacting climate change, as I can with no confidence say that it's not impacting. Okay, the physics behind it says there is an impact. What we don't know is how big of an impact. Okay, because there's a lot of mitigating factors. But if we are in a changing climate and certain individuals are getting stressed, there is this natural selection process going on for better adapted individuals. And this is where, some people say, well, we ought to do assisted migration, where we take trees from warmer latitudes and move them to colder latitudes because we're warming. I don't like that theory at all.
Acorns.
Yeah, I don't like that at all, because when you think, okay, it may be viable in certain areas, but in the northern Rockies, these species have been here for a long time. There is a lot of genetic depth in them. Not maybe in an individual, but in a population. There's a lot of genetic variability that's there. What is happening, what will happen is those that have the characteristics for this climate will be selected for, and so there is a natural process of selection going on there. In the case of lodgepole pine, because fire tends to slow that down, what we could potentially do, again, theoretically, is now if I were to go in here and harvest the dead stuff, leave the live stuff behind, remove the seed source that the fire will promote, and leave a dominant seed source that the bugs have promoted, then I'm doing what I might call assisted adaptation. I'm assisting a natural process move faster. Okay? So this is perhaps the direction that genetics might be moving in all this stuff, where we used to say, okay, here's the biggest, tallest ones, that's what we want. Well, if I'm growing wood, fine, but then I'm going to have to use pesticides and all this kind of stuff. I mean, and it's worked wonderfully. Monsanto is a prime example. Okay, what did Monsanto figure out? Okay, they created Roundup, which kills everything, all vegetation, and then they genetically modified wheat to be resistant to Roundup, the mechanism of Roundup. Okay, so how do I become a multi-billionaire company? Okay, I make a solution, and I make even a better solution, so I sell both of them. It's so successful that a test of flour worldwide found that 90% of wheat flour sold worldwide has traces of Roundup in it. Okay, so again, now is that a bad thing? Well, we can't grow enough food to feed our population, so it's a good thing because we got a lot of hungry people that we got to feed. As a matter of fact, the latest USDA estimates for Montana indicate that Montana will have to produce 3 times as many cows as it does now to meet the global demand for beef. Okay, is that possible? Not with the cows we have now. So, maybe we're going to have cows that don't move and they just get big, and, and you hook them up to an IV. Who knows? Okay. Hard to argue with people who are hungry. Okay, and that's getting off on a different tangent. But with trees, there's where we used to select for fastest growth. What we might want to be selecting for now is robustness and resilience, greater genetic diversity and base that has a greater chance of surviving as a viable population in a variable climate, or with introduced diseases, or on and on and on. Okay, so our knowledge of genetics has given us our improved knowledge of genetics has given us this ability to look at things that are beyond just the simple, okay, if A then B, and I'm going to get this kind of a return on it. Now, how does that affect you as a forest landowner? Why does this make a difference? Well, all of us, go through here. Well, one, I'll stop. Okay. I just wanted to show this, that these kind of phenomenon happen over and over again. This is eastern Montana, ponderosa pine forest on about 10,000 acres of national forest. Craft Springs, pre-Brewer Craft Springs fire. Brewer fire killed about 30% to 60% of the trees where you were, created a pretty high fuel loading. It burned again because of all that fuel loading. And what happened was, this is now all that's left of that forest. Okay, so now when we have these little isolated populations, what's going to be happening to the genetic variability in the trees? We're going to probably see a high degree of inbreeding again occur across this landscape. This is what has happened historically across northern Rockies. Okay, populations shrink, they swell, they shrink, they swell, and we get this genetic degree of inbreeding in there, and there's some kind of correction factor that occurs, and we go back and forth. You see this phenomenon anywhere, so here on our varied topography, we have Douglas fir, sagebrush, Douglas fir, sagebrush, pretty hostile environment in between here. Chances are that each of these populations in here is terribly inbred because they originate. You can go in there, you find 3 or 4 matriarchal, big old matriarchal 500-year-old Doug fir in there, and the rest is surrounded by this younger stuff that may be 100 years old. And all of a sudden spruce budworm goes in there and decimates most of it. But not all of it. You'll always find some that are not being hit. Okay? This is how these processes work. So now, if you're a landowner, skip past that, and you're making decisions out there, okay? You're doing thinning for your objectives, wildlife habitat, wildfire hazard reduction, things like that. Historically, the guides you get is what's called a Girard growth form, where you see the tree crown and you're selecting for the biggest and the tallest, right? Well, with what we've learned at this point, which is admittedly quite incomplete in knowledge, how do we change that? Okay? When we look at, here's a ponderosa pine regeneration in this opening. Okay? Ponderosa pine seeds do not fly with the wind unless it's a hurricane. Okay? The cone lands on the ground and it rolls and the seeds fall out. Chances are that the majority of these ponderosa pines are all full siblings. They're all related to each other. Okay? Which means there's also a certain degree of inbreeding that occurs in there. Now, from what we hopefully have learned out of that, historically, when I were to thin this, what trees would I leave? Joe, which trees did you leave?
The older trees,
How do you know they're older? The bigger trees, right?
the bigger trees.
So I'd go in here and say, oh, well, this one is growing gangbusters, and that one's growing well, and that one's growing well, and I'll just get rid of the rest. Well, if we're only selecting using height as our selection tool, we're leaving homozygous inbred ones. Okay? At the same time, in breeding depression, the most common phenomenon is dwarfism. Okay? So we have all these little ones in here. And there's a chance, a higher probability, that they are inbred as well. So with this new knowledge, what we want to be selecting for, if all this stuff holds true, and again, species are going to vary by species, okay? Just because this is what's working with Norway spruce, doesn't mean that this is how ponderosa pine is operating, though it stands to reason that it is, okay? What we want to select for are perhaps those that have the best crown by the best needle retention, the best needle color, the best, what might be described as robustness of seedling. Because what that tells you is if it has good color, it's getting the nutrients it needs to produce adequate chlorophyll. Okay? And it means it's physiologically doing best on that site, and height growth isn't an indicator for that. And our chances are then that we're leaving those that have the highest genetic diversity in them. So that's our new selection criteria that we might want to use to leave the best out there. Now, again, the same thing holds towards cereal crops. Original grains, kamut and all those things have a lot of genetic variability in them. And again, that genetic variability means they produce lots of different proteins and different kinds of sugars, okay, which has been found to be better for us. If you take that and you breed it to only produce glucose and not arabinogalactose and all the other sugars that are out there, it'll get bigger and have a little more starch in it, and we get supposedly more food value. But, you know, as a nutritionist once told me, we're starving our youth to death. They're obese, but they're starving to death because they're not getting the variability that comes out of these old grain crops. This same type of philosophy also works with trees. If the purpose of when we leave a tree behind, and we're doing a selection process, we want to leave trees that are going to live long, right? That are going to grow. Unless we're Weyerhaeuser, that's growing a tree in 30 years and we want it to be 20 inches in diameter. It's a different objective. I have no problem with that, okay? South America, like in the southeast, they grow loblolly pine to salt log size in 30 years. Chile and Brazil have a genetic modified version of loblolly pine that grows to 20 inches in diameter in 15 years. Okay, they're grown like corn with the mill in the middle. Okay, it's a great way to produce a lot of fiber in a short period of time. But if that's your objective, great, grow hybrid poplars. Okay, same type of principle behind that. But if your objective is to leave trees that, you know, in this case, is a very good friend of mine's property. He thinned this 18 years ago. And, you know, if you get the Forestry Spring newsletter, you'll be familiar with this picture because it's right on the cover. And the question is, okay, should I thin this? Okay, well, what are your objectives here? I mean, you got to look at growth rates and everything else, but are there key characteristics that you might want to look at in there? This, and yeah, he said, yeah, let's thin it. This is the result. So what are the advantages? Individual trees might be getting more of the nutrients and the water that they need. But when you're selecting, if you remove trees to a certain sweet spot distance, there is a chance that your trees will be pollinated by other families of trees, and therefore the genetic variability will be maintained. Okay? Whereas trees that are next to each other tend to pollinate each other. So the closer they are, the greater the likelihood that they're related, but the greater the likelihood that they're pollinating each other too. Now, this backfires if you open up those distances too far, because there's a certain distance pollen won't effectively travel and be viable. Okay? So, and it's found if you leave one tree on this ridge and one tree on the other ridge, they won't pollinate each other. They'll self-pollinate themselves because it's too far. So what that sweet spot is going to, again, vary by species, and we don't have good answers for that. But there's other characteristics that you might look at on there. Crown characteristics, okay? I already mentioned height growth is actually negatively correlated to, in Norway spruce, to genetic variability, okay? But, you know, of course the tree has to live, so, you know, some trees, here's two ponderosa pines. This one has nice, thick needle retention. This one has poor needle retention. It's suffering what I call a Q-tip effect, where the branch ends look like Q-tips, because there's not a lot of needles, they don't retain them for a long time. Is that genetic or is that environmental? You can't tell. You don't know, okay? Like I said, it would be nice someday if we had a little probe and we just stick it in there and it'll tell us, okay, that, oh, this one has a lot of genetic variability for the specific markers that we're looking for, and this one doesn't. We don't have that right now. I can collect samples and send them to a lab in Vienna, and they'll do it for me, okay, but that's not going to help me in my marking prescription. So there, you know, you got to look at the old things, growth rates. These trees were slowing down. So, you know, here's a release 18 years ago, and here's where we are now again. Okay, so back to the question, should that stand be thinned? Well, if you want to have trees that are a little bit more resilient to fire and have all the energy they need to, if they have the capacity, defend themselves. Other crown characteristics you might look for other than height growth. Well, years ago, crown characteristic guides were developed for foresters marking trees. One of the things that was selected against was these angular branches coming off the ponderosa pine, okay, versus horizontal branches. The reason for that was because when these grow into the wood, you get long knots and a lot of wood defect, okay? However, I've been observing trees now for 30 years. I never get bored, and my wife yells at me because I'm looking out the window at trees while I'm driving down the highway. But it seems, again, genetics can vary a lot, and you can have local nuances. On my property and surrounding areas, and in the Bitterroot, I've noticed that trees that have these ponderosa pine trees, that have these vertical branches, tend to suffer a lot more from needle cast issues. So there are genetic characteristics that may be linked together. Okay, many of them are linked together. Okay, polymorphism. Okay, it's not just one gene determining something. There's a whole host of genes that are doing stuff. And I found that most of my ponderosa pines that have these vertical branches are suffering from needle cast disease, and the ones right next to them are not. Okay, if you have,
Is that on your property?
yes,
Are those two trees pretty close to the same age?
yeah. Yeah, one's a little bit younger, but yeah. But I see that on other places as well. Again, one observation doesn't mean anything other than observation.
Right.
And that's, you know, as a landowner, the best thing you can do is constantly being out there and looking for stuff and trends and tendencies. because in science we like to average across broad landscapes. The problem with averaging is you miss a lot of good stuff, okay? And again, when you look at the Northern Rocky, every valley can be genetically different because of the flow of air and pollen spread. And then also think about that all the trees you're seeing originate from tiny little populations 2,000 years ago. So you may be looking at the 5th generation of trees, okay? That is at the height of inbreeding depression if it originated from a small population of trees, okay? So, I mean, this, when I started thinking about this stuff, it kind of blew my mind because I used to, when I first came here, thought, well, these endless forests were here forever. Well, they're a recent phenomenon. And we're suffering the consequences of those small populations repopulating large landscapes, okay? So from a genetics perspective. So that's one thing. Height growth, as I mentioned, you know, if I look at these, okay, well, this one's growing taller, narrower, this is growing broader, wider. Are there any inferences that I can make from that? Well, not with a lot of confidence. But in ponderosa pine, another thing I've noticed a lot of is the ponderosa pines that have the biggest internode growth and are growing the tallest, fastest seem to be the ones that mountain pine beetle hit first. And I've suffered this on my own property, where I took out the short squat one and I left the two next to it that were growing gangbusters, and 3 years later mountain pine beetle killed them. I was pissed because I wanted those trees, you know. So, I mean, again, it's something you got to look at, and observation can be very biased. Agree, you know. So, you know, is this a good tree to leave? You know, look at that internode growth. But the question is this environmentally caused or is this genetically caused? And so this gets kind of dicey on there. Let me scooch on. Can you see this phenomenon on younger trees? You know, we already talked about that. You know, you got to look at what's around there. If there's only 2 ponderosa pines that have repopulated all the regeneration in there, you know you're going to have a lot of inbreeding going on in there. If it's a big ponderosa pine forest, maybe not. Here's one that I've talked about before, top kill on ponderosa pine by Ips. Okay, Ips has 2 generations in a year. In the spring it's in the slash. Later in the summer when the 2nd generation emerges, a lot of these trees are water stressed, particularly the tippy tops of trees. And so it can infest the top of a ponderosa pine because it's suffering from drought stress and it doesn't have the water pressure to produce pitch up there to pitch them out. Interesting, another theory is that when we talked about this, or someone brought this up earlier about whether genes can be turned on and off. Okay, well, it's been shown that they can be by certain stimuli. Okay, but it's not uniform. It varies among species and it varies among individuals. So I'll throw an observation at you that may be totally wrong, but nonetheless it's one I've observed. And this goes back to my previous example, that when ponderosa pine is injured, it seems to shift into a defense response. And there's some fundamental hallmark biological work called Defend or Grow, was the name of the paper. Okay? And that is that organisms do have this capacity, some of them, to shift where they allocate their energy. Trees in particular, because a response to crowding is for trees to grow taller, okay? Because they need to get to light. A response from, particularly ponderosa pine and larch, to injury may be to shift allocation to defense. Because what I've seen is top-killed ponderosa pine tend to be the only ones that survive subsequent mountain pine beetle outbreaks. And ponderosa pines are great survivors. They're dinged by equipment, whatever, they survive. Now the same thing for Granford Doug fir. Once you see that on Granford Doug fir, within 5 years it's dead. They don't have that ability. So it varies by species. Now I tried to talk to the landowner leaving this tree. He had none of it. He said, no, this one's going. Okay, but again, that's fine. There is nothing set in stone in all these things. So what you get is genetics plus environment, plus the interaction of genetics and environment. That's the age-old formula that's out there. Now I'm going to add, what about, can that be altered by stimulus and genetic depth or heterogeneity? The more wheels or heterozygosity an organism has, the greater capacity it has to respond or react or defend itself against some kind of pest or pathogen, is the theory. And it's, you know, you find it across a broad spectrum of genetics and organisms. And so again, you know, the bottom line is when we're selecting on things, that shouldn't guide us, but we should think about it. It should be in the back of our mind at all times. And so in this case, you know, it's pretty clear that there's some differences going on there. And this variability can occur in smaller. This is all grand fir, and this frost, super cold November, hit that one, but not the ones around it. Shows you the genetic variability that's there, and that genetics may also exist within a single organism. So here's one grand fir, and this part got hit by that late frost, or that super cold, and the rest did not. And you might argue, well, okay, it's got this asperon, which is providing a thermal effect, so these branches had not hardened off as much as that. And this phenomenon has been found in eucalyptus. Remarkable that in a eucalyptus tree, every branch on that eucalyptus may have different genes turned on, producing different terpenes and defense mechanisms. It was in a recent paper of Nature, and it's blown people away to see such variability in one single tree. And of course, the adaptive mechanism is they can be defoliated by koalas and other organisms, animals. And so by varying their genetics in the branches, producing different terpenes and defense mechanisms, they're ensuring that some part of the tree is going to keep leaves on it. It's a survival mechanism that's been selected for across a long period of time. So, you know, this goes back to, you've got to observe. And, you know, there is no simple guideline for when you're looking at your forest, what do I leave? But now you got more to think about, you know. And, you know, I joke because I have 20 acres and I'm getting ready for another thinning, another load of trucks, another truckload of logs I'm going to cut out of there. And believe it or not, I take about 6 months to decide which trees I'm going to cut and which ones I'm going to leave. Now, if I'm a production forester, I can't afford to do that. But, you know, you think about species, think about variability within the species that you have. So, questions? Yes, sir.
Would you leave one of those top kill trees on your property?
Oh, I've got 3 of them.
You do?
Absolutely.
And how many years have you been watching them?
10 years.
And you're
They're still there.
empirical, and you don't think it's spreading to the trees around it, or can you tell?
Well, okay. There's a difference if it's a disease or an insect. Okay. So Ips.
Let's
hits
say
it.
it's a pine beetle. I mean,
Okay, yeah. Well, it hits a tree, it's out. It's gone. Okay. What the beetles are telling me is a certain susceptibility, either that individual or that part of the tree. Okay. So that dead top is not going to infect other trees. Okay. Now, if it's mistletoe, that's a different thing. Then I have an infection source. So the Ips top killed this tree. The growth rate on the tree, if I'm trying to grow diameter, is deplorable. I mean, they're 40 rings to the inch. It's almost non-existent. But in the meantime, I've been fighting western pine beetle. That's what my nemesis up in Evero. I've been fighting it for 20 years. And every year it comes and kills a tree or two, and the worst, it killed 10 trees in 1 year, and including some that I really wanted to leave. Okay? That tree has not been hit. Another tree that was hit by lightning has not been touched. Even all that pitch on there, all this wonderful, you know, attractant that's there, an injured tree, has not been hit. And so my wife was after me. There's one that the Ips killed everything except for that last world green branches. She says, "Aren't you going to take that tree out?" I said, "No, this is my experiment. I'm watching it." You know. And see, I work in windbreaks and shelter belts a lot too. Trees, particularly ponderosa pine, can suffer an enormous amount of damage and stay alive. I see it on windbreaks and shelter belts all the time. You go, how on earth can this thing be surviving here? You know, but they don't grow fast. You know, and so, you know, the needles are nice and green, and you know, my objectives are more than just timber production. I don't mind the money, but it's not going to make or break me on 20 acres. But I want that tree there, and you know, the top of it is for cavity nesters, you know, chickadees and whatever, and we'll see. But I used to mark trees for Boise Cascade out of McCall, Central Idaho, and boy, I've got into some serious arguments with my bosses down there because, you know, we're looking at big trees and little trees and things like that. And their rule was anything over 20 inches went. And I came across this magnificent ponderosa pine. It was 4.5 feet in diameter, 50% crown, full, just, you know, one of those trees. And boy, I painted that thing red, which meant leaf. you know, and I painted it really red because I knew it was a big temptation to take, because there was 5,000 board feet in that one tree, okay? And my boss, a week later, or a month later, came in the office, and I knew something was up because he looked pissed. I was out there on a logging job, and he'd come across this tree, and he came in the office, and he just chewed me up one side and down the other, and said, you know, we can't afford to leave trees like that. At that time, I said, well, you can't afford not to leave trees, because that tree's a genetic reservoir right there. You know, it's survived. I mean, I cored it and estimated it at 300 years of age. I said, it's been there for a while. It's survived all of these different things. And so my guess is it has the genetics, superior genetics for that site. And that's what you want repopulating the area. You know, now that was, you know, I was, had a master's degree and I just finished all my genetics coursework and was thinking along those lines and basically didn't know for sure one way or the other. But, you know, when in doubt, diversify, right? So, but I've been watching this for years and years and years, and I'm trying to get a research project going with the University of Vienna that does all this gene mapping. And all it requires is doing some surveys to these beetle hit forests, collecting a sample from beetle hit trees and from live trees, and testing for certain, you know, you can just use 8 or 9 allozymes as markers, and seeing whether there's a difference in genetic variability. In Norway spruce, it's huge. And so, and we could start, you know, maybe changing some of how we're looking at what we're doing out here. And to me, the proof in the pudding is with this pollen analysis. When that came out, and we see how young our forest landscapes are, and they originate from tiny little populations. So, you know, those are all the pieces are there.
So what was the final on you can't afford not to leave a big tree?
It got cut. Boise Cascade shut down all its mills 10 years later because they had no volume left.
Yeah.
It's sad. There were 5 mills south of the Salmon River. There's none now.
But that was really one that should have been left. Can't
Yeah.
afford not to leave.
Yeah, and it wasn't like we were leaving, you know, 1,000 acres of big trees like that. This was one tree on a ridgetop, you know, in a drainage, you know. So, but, you know, I understood my boss. He was under pressure to cruise volume. You know, our mill needs X million board feet per year to stay open. Yes?
My family started planting, you know, in areas. Do you recommend that? Planting seeds and
Certainly, because what you're getting is the genetics. Well, it depends from where, of course. If you're buying from the state nursery, they have their own seed orchard for western Montana pine. Sometimes they buy seeds that are collected from different places. So you're introducing some genetic variability into your local stand, which I used to think, well, leave your local stand alone because it's been selected for and it has the genetics it needs for that site. The thing about Northern Rockies, like, you know, this last couple of weeks is if you don't like the weather, wait a while. You know, it'll change.
yeah.
So we have we're defined by variable weather. And so maintaining some genetic variability and hence resilience is good. So yeah, I mean, bringing something in is not necessarily a bad thing. You know, we have a lot of Black Hills ponderosa pine planted in Montana because in the 40s and 50s, if you've been in the Black Hills, you know, you see Black Hills ponderosa pine, these scrawny, branchy things. You know what I'm talking about up in the Sealy Swan area. You know, the Black Hills are magnificent. And so I could see why they selected those for the genetics in the Black Hills, which is a more humid environment than here. Here, they're branchy, they're short, they don't grow real tall. And so there's actually some people who called for eradicating all the Black Hills ponderosa pine that were planted. Well, if your objective is to grow tall timber-producing trees, yeah. If your objective is to increase genetic depth and variability into the landscape, then leave them. Because if they're really maladapted, they won't reproduce and they won't survive.
Right.
And so, and we see that in windbreaks. You can get ponderosa pine to grow up in Chester and Hever, but they never reproduce because their seed cannot survive on those harsh conditions. And there's a term for that, it's called exesis. Is that the true test of whether a species is adapted to a site, whether it successfully reproduces and its offspring successfully reproduces. So I mean, once, you know, there's a movement to eradicate all windbreaks and shelter belts across Montana because these conifers are polluting the landscape out there. Well, you know, with the exception of Russian olive, they're not going anywhere because they can't reproduce. So I don't know if that answered your question or not, but
It was just a thought, you know.
Yeah, well, always check where your seed source is coming from. And, you know, again, most selection guidelines have been for tall, big trees. That's what you use. But when they buy cones from people, what they're looking for are from healthy stands. And so there's a lot of variability into that. Yes, you had a question, sir?
No. Well, I was gonna ask you, the one with the vertical limbs
Uh-huh.
there, that was the one you would cut?
Yeah. I've developed a discrimination against them just because every single one that has those characteristics on my property has needlecast issues. And when I see something and I look at a broader landscape and I see very similar characteristics in the bitterroot, I mean, the bitterroot is a weird example because the bitterroot used to be a very dry valley. But now with people moving into it, everybody has a lawn they water. There's center pivots, there's irrigation. So you go out into the bitterroot in the end of July, and you get up high and you see this haze in the valley bottom. And it's the humidity from all the air. Well, that humidity changes the environment for fungal diseases. And there's a reason that electrodermal needlecast is going ballistic in the bitterroot, in my opinion, because now you have a humid environment on a tree species that's not adapted to a humid environment, and more or less it's not adapted to fight off the pests and pathogens that survive in a humid environment, like needlecast diseases. White bark pine has very little resistance or resilience to pests and pathogens. Why? Because it grows in one of the most harshest environments in the earth where fungal organisms and pests and pathogens can't survive. It doesn't need it. Okay, so it was never selected for that characteristic. So anyways, you got to get lunch. It's kept you 10 minutes longer already. Thank you. Lunch is over, you know where, over in the student union building?
Same place as last year?
Yeah. So thank you for your attendance.
Thank you.
I hope this was helpful.